Hello, welcome to BioWise with Aanchal. How are you all doing? I hope the preparation is going on well. Now today we will be talking about the topic basic biology. Now the basic biology comprises of two chapters, that is your chromosomes, the structure of chromosomes, cell cycle and cell division, and genetics. The entire paper may have around 12 to 14 marks. Stay connected till the end to know and explore the content of basic biology. So I will be talking only the important aspects of this unit of your biology paper, which is important for the exams. Okay, so let us begin with basic biology. How many marks it comes for? It comes for around 10 to 12 marks. Now children, before we understand the concept of cell division, cell cycle and your structure of chromosomes, you need to know these words. Right? Chromatins, chromatid and chromosomes. And then one more is there, sister chromatids. Okay, so when we talk about chromatin first, what is chromatin? Chromatin is a thin thread-like structure which is present in the nucleus of a cell. Okay, so it is a thin thread-like structure. What does your chromatin consist of? It consists of your DNA and the histones. Okay, then what are chromatids? Now what happens during the cell division? These chromatin fibers condense to form chromosomes. So chromosomes, if you see, has this kind of structure. Okay, so when you split it into two, okay, the point of attachment of the chromatids is centromere. Okay, so this is called a chromatid. This is called, the entire half is called the chromatid, and these two are called the sister chromatids. Okay, these are the sister chromatids. Chromosomes, what are chromosomes? The entire structure is called a chromosome. And sister, I think I could explain you what is chromatid, what is chromatin, and what is chromosome, and what is sister chromatids. Now you are aware that we inherit one chromosome, you can say, from the father and the other chromosome from the mother, right? So let me quickly wipe this out. I am just talking about the basics, the conceptual understanding, so that you can write your paper well. Please don't mind the color, the blue and the pink, the traditional ones. Okay, the two chromosomes of same size, shape and the composition is called a homologous chromosomes. What it is called? Homologous chromosome. Okay, just suppose it has been inherited from the father, it has been inherited from the mother. These two are the sister chromatids. The chromatids has duplicated and it has made a sister chromatid. These two are the non-sister chromatids. Okay, non-sister chromatids. So what happens when they come closer to each other? That is synapsis. Okay, synapsis. There is another word called synapse. Remember, this is the neuron junctions we have studied in the nervous system chapter, and here synapses, that is coming closer, that is homologous chromosomes come closer to each other for the process of crossing over. So that is called the synapsis. Now what happens? These two cross their wings. Okay, these two. So this structure, this X-shaped structure is called chiasma. Okay, and the process is called crossing over. So understood the concept of synapsis? If you just want to understand, like two friends are coming together. So coming together, shaking hands is synapsis. Taking a photograph of shaking hand is chiasma, and exchanging gifts is your crossing over. Is that clear? So afterwards what will happen? After the crossing over has taken place, you will have the recombinant chromosomes, and this recombinant chromosomes are the cause of variations, and this happens in meiosis. Okay, is this concept clear? Yeah. So I hope you have understood the basic terms. Now we will go a little bit more in the chapter. You know that chromosome of human beings, chromosome number 1 is the largest chromosome, and chromosome Y, that is present in human males, is the smallest one with the fewest number of genes present in it. Yes, children? Now let us understand the basic components of our DNA. Nucleic acid. Okay, there are basically two. You know that full form can be asked in the examination: DNA and RNA. Okay, the nucleic acid contains sugar, phosphate, and the nitrogenous base. Okay, so in this case, DNA and RNA, DNA contains the sugar, which is called deoxyribose sugar. It contains the phosphate group, and it contains the nitrogenous base adenine, guanine. These two are purines, and thymine and cytosine, that is your pyrimidine. Okay. In case of RNA, the sugar present is ribose sugar, same phosphate, and the nitrogenous base present here is adenine, that is guanine, and cytosine. It remains same as DNA, and instead of thymine, RNA has got uracil. Is this clear? So this is your basic components that we need to know. The basic unit of DNA is a nucleotide. What is nucleotide? Sugar plus phosphate plus nitrogenous base. Okay, nucleotide. And what is a unit of chromosome? Chromosome is nucleosome. What is nucleosome? Nucleosome is DNA plus histone proteins. This much if you know, is more than enough for the examination. You need to know from the basic unit. The basic unit is nucleotide. Nucleotide combines to form a DNA. Then DNA will be coiled around the histone proteins to form a nucleosome. Nucleosome will wind up, will coil, supercoil to form solenoids. Solenoids will form the chromosome. Okay, so understand this. You can take a screenshot if you want. Let me quickly rub it. So I hope the concept is clear about the nucleic acid, its component, what is a nucleotide, right? Watch the video till the end for some important questions that we will discuss, what is generally asked from basic biology. And if you like it, please subscribe it. So let us move ahead. Now next one is your cell cycle. What is a cell cycle? Cell cycle, as the life cycle of a cell, cell starts to grow and then divide. So you have to make a circle. It's a ring with a diamond attached to it. What are the different phases? You remember PMAT, that is prophase, metaphase, anaphase, telophase. And after that you have cytokinesis. And what are these? These are karyokinesis. Okay. And what is this? Mitosis. And then you have the phases. Let me just quickly go through the phases. This one is the G1 phase. This one is the S phase, and this one is the G2 phase. Okay, this phase, that is from here to here, is the interphase. Okay, after interphase you have the mitotic phase or the mitosis. G1 is called the first growth phase. Followed by the synthesis phase. This synthesis phase is very important. Synthesis phase. It has been asked in 5 years, I think it has been asked 3 to 4 times. So synthesis phase, and this is the second growth phase. Now if there are certain cells like neurons, nephrons, they do not divide. Okay, so what they do, they slip into G0 phase or G0 phase, that is the resting phase. Okay, the resting phase. Is this clear? So what happens in the G1 phase? The necessary proteins required for the DNA synthesis and more RNA is synthesized in G1 phase. The cell increases in its volume, obviously, to be prepared for synthesizing more and more DNA. That happens in the S phase. So S phase is for DNA synthesis. S phase is for DNA synthesis. This is very important. S phase is DNA synthesis. Growth 2 phase mein you need all the mRNAs and the proteins required for the mitotic division. When you talk about the mitotic division, there is karyokinesis and cytokinesis. Karyokinesis is nuclear division, okay, and cytokinesis is cell division, that is cytoplasmic division. Okay, this is what the cell cycle is all about, and that you must know. You can take a screenshot. Is that clear? This was your cell cycle. Now we will talk about the mitotic division, because in this part of the chapter many questions are asked on the phases of mitosis, either to identify the stage or draw the next phase. You draw the phase which follows the diagram, you have to draw the phase which precedes the diagram. Precedes means the step before and follow means the step after. So you have to remember there are 4 phases: PMAT, prophase, metaphase, anaphase and telophase. The cell generally stays in a resting phase, that is the interphase. In the interphase, the cell stays, there is a nucleolus, the chromatin fibers, right? This is what the cell looks about. This is your interphase. After that, the cell enters the prophase. In the prophase, the nuclear division, that is the nuclear membrane, starts to disappear. the chromosomes becomes thick. The duplication has already taken place, children. So here the chromosomes just becomes thick. Okay, I am just drawing the cells with the 4 chromosomes. So now the cells have become thick and the centriole starts moving to the opposite pole and the spindle fiber begins. Okay, this is what the prophase is all about. You have to label the diagram. Label is easy. You have the cell membrane. When they ask you to identify it as an animal cell or plant cell, look at the cell wall and look at the aster formation. There is no aster formation in the plant cells because plant cells lack centrosomes, centrioles. So there is no aster formation taking place in the plant cells. That is a point of identification whether the given cell is a plant cell or an animal cell. Fine. Then the nuclear membrane disappears. That is the nuclear membrane, and even nucleolus disappears. Yes. Then you have the cytoplasm. You can label it, the chromosomes, the centromere. You can have 2 to 4 labels, that will be enough. So this is your prophase. Yes, after prophase, next phase is metaphase. So for metaphase, you draw a circle, the equatorial plane. Okay, you have to draw 4 chromosomes because we started with 4. Kindly maintain the number of chromosomes. Okay, and this question is also asked, how many chromosomes the cell has. Do not write 46 human chromosomes, write about the chromosomes present in the cell. Okay, so this is how the chromosomes are arranged in the equatorial plate, and this is your metaphase. I am not labeling it, children. I hope you know how to label it. This is your metaphase. The chromosomes aligns on the equatorial plate. M for metaphase, M for the middle of the cell. So all the chromosomes are aligned in the middle of the cell in the metaphase. Next one is the anaphase. That is the chromosome splits. The chromosome splits to, right. So what happens? The chromosomes split. The chromosomes moves away from each other. A for anaphase, A for away from each other. Right. So this is how you identify if the chromosomes are facing the wings are facing each other and they are moving up towards the opposite pole. This is anaphase. Anaphase can be included in 2025 examination. Can be, okay. You have to go through all the phases, that is important. After anaphase, what happens? There is a small furrow formation begins in case of the animal cell, and the nuclear membrane starts to appear. In prophase it disappeared, here it will appear, and the chromosomes are totally separated at the 2 poles. Yes. So chromosomes, again their chromosomes will start becoming thread-like. Okay, and you can see here the nucleolus and all will start appearing at the later part of it. This is what telophase. This is telophase. So after telophase constriction, reappearance of the nuclear membrane, nucleolus also reappears and the chromosome starts becoming thread-like again. So this is what your 4 diagrams are. This is karyokinesis. This one is karyokinesis, that is nuclear division. Okay, so differentiate between karyokinesis and cytokinesis can be asked in the exam on the basis of their definition. So karyo is nucleus, kinesis division. Division of nucleus is karyokinesis. It includes the PMAT, that is your prophase, metaphase, anaphase, telophase. Is this clear? I'll just rub the prophase to explain you cytokinesis. After karyokinases, we have cytokinesis. In cytokinesis, what happens? This furrow deepens and you have got two separate cells. So, this process of separation where two different cells, daughter cells are formed is called cytokinesis. In cytokinesis, the cell resembles the cell present in the interface. Okay, that is the nuclear division followed by cytoplasmic division has made two cells, two daughter cells, same as the parent cell. The two daughter cells which is diploid. So, two diploid daughter cells are formed after one mitotic division. Is this clear? So, what is the significance of mitosis? Grr. What is the significance of mitosis? Growth, repair, replacement, reproduction, asexual reproduction. So, what are those? Growth, repair, replacement and asexual reproduction, right. So, what is the outcome of mitosis? One cell give rise to two diploid daughter cells. Is this clear? This is the first part of basic biology. Next, we will be talking about genetics. Meiosis, crossing over, we have already discussed. What is crossing over? What is synapses? What is chiasma formation? The difference is clear, I guess. If not, put it in the comment section. I will explain you again in the comment section. Yes, significance of mitosis, GR, that is growth, repair, replacement and reproduction, asexual reproduction. What is the significance of meiosis? It is production of gametes, that is reduction division. So, meiosis is basically a reductional division where the number of chromosomes is halved in the daughter cells. So, after one mitotic division, you have two diploid daughter cells formed. After one meiotic division, you have four haploid daughter cells produced, basically the gametes produced. Oogenesis, spermatogenesis is possible only because of meiosis, and it is only because of meiosis that the chromosome number in a species is retained, because for every gamete formation the chromosome number is halved and then again on fusion it restores the original number. Is that clear, the significance and all? Any doubt, put it in the comment section. It will be replied, children. Now, next we will talk about genetics, the next aspect of basic biology. Next part of basic biology is genetics. In genetics, what you must know is the father of genetics, who is Gregor Johann Mendel. Gregor Johann Mendel, the father of genetics, and the plant he worked on, which is Pisum sativum. Pisum sativum. When you write the scientific name, you need to underline the same. Okay, scientific name must be written with the first letter capital, second letter small, and the word should be underlined. This is the common pea plant. Now, Mendel studied certain characters which he observed as pea plant was tall and the other trait was dwarf. Then he saw that the color of the seed was yellow and the color of the seed was green. Then he saw the color of the pod. This is seed color. Seed color. This is the height of the plant. Okay. Then he studied pod color. Pod color was green and yellow. Then pod shape. It was inflated and it was constricted. Constricted. Okay. Then the flower. Flower position. Flower position was axillary and terminal. Then it was flower color. Flower color was purple and white. Okay. And the last one, the seed shape. That was round and wrinkled. Now, these 7 traits were studied by Mendel. What you need to know is none of the letters are repeated. Tall, axillary, yellow, green, inflated, right? So, you can see the alphabets are not repeating, and the names written on this side are the dominant traits, and the written on that side is a recessive trait. So, we use the first letter of the dominant trait. So, for height we have 2 alleles, that is capital T and small t. For yellow seed you have capital Y and small y. Green pod color, inflated and constricted, axillary and terminal, purple flower and white flower, round seed and wrinkled seed. So, now we have to know what is dominant and what is recessive. So, the traits which were able to express themselves in any condition, that is homozygous condition or heterozygous condition, is considered to be a dominant trait. And the trait which can express only in a homozygous condition, like the small t and small t can give rise to a dwarf character, it is a recessive trait. Is that clear? Homozygous means it contains same type of alphabet. This is homozygous, this is homozygous, but this is heterozygous. Is that clear? Homozygous, heterozygous. This is the character what we have studied, and you need to know because cross can be asked on any of these. Generally, the monohybrid cross is asked. Dihybrid cross, phenotypic ratio is important. Okay. So, now let us quickly move ahead to the monohybrid cross. The next topic is the monohybrid cross. As I will be talking about the monohybrid cross and the dihybrid cross, I will talk about the laws of inheritance. The 3 statements given in your textbook for the laws of inheritance, law of dominance, law of segregation and law of independent assortment, must be written the way it is written in your textbook. So, this has to be learned. So, monohybrid cross. Monohybrid cross means only one character is studied. Let us study height. So, height is a character. Alleles as traits. So, traits are the different forms of the character and alleles are the alternative forms of genes. Is that clear? Now, monohybrid cross we are studying about the height. So, height, let us take the parents. This is the parent. Parents are the purebred lines. Purebred means they are homozygous tall and they are dwarf, homozygous dwarf. So, this one will give rise to the first generation, that is F1, the first filial generation. Before that, you need to form the gametes. So, in this case, the gamete formed by a homozygous tall plant will be only one kind, that is capital T, and here it will be only of one kind, that is small t. So, the F1 generation will be capital T, small t. Is this correct? So, this is a heterozygous tall plant. Now, this explains the law of dominance. What the law of dominance states? Out of the 2 contrasting traits of a character, the one which expresses itself in all conditions is the dominant trait, and the one which is suppressed is the recessive trait. This is the law of dominance. Out of the 2 contrasting pair of traits of a character, Is that clear? That's the key word. Any doubt? Now this one is the F1 generation. F1 generation of a monohybrid cross explains the law of dominance. Next one is the law of segregation. Segregation. Now in law of segregation we have already seen that F1 was capital T as small t. So let us self F1, okay, selfing of F1. That means capital T small t cross capital T small t. Here I will introduce the concept of Punnett square to you. Here the gametes formed will be capital T small t in this case and capital T small t in this case. Punnett square is a kind of square drawn where, please, it's a square, it has to be a square. Make a square and then put the gametes on the top to find all the possible combinations, okay, all the possible combinations. So here what is the possible combination? Capital T small t, capital T small t and small t small t. In this case the reappearance of the dwarf trait in the F2 explains the law of segregation. Law of segregation states that these two contrasting factors separate themselves in the gametes independent of each other. So you can see that this is also going into a gamete, this is also going into the gamete. So they just move into the gametes without influencing each other. They separate themselves into the gametes independent of each other. Okay, so this is your law of segregation. So what is the F2 formed here? What is the F2 formed here? You can see there is capital T, that is your homozygous stall. Then you have 2 capital T small t, that is heterozygous stall. And 1 small t, that is dwarf. Dwarf is a recessive trait, so obviously it has to be homozygous. So this 1, 2 and 1, this gives you the genotypic ratio. Genotypic means the genetic constitution. The genotypic ratio of F2 in a monohybrid cross is 1:2:1. Okay. Genotypic ratio of the F2 generation of a monohybrid cross is 1:2:1, and the phenotypic ratio, though it is homozygous, it is heterozygous, but the output, that is the physical appearance of the plant, is tall. So phenotypic ratio is 3:1, where 3 is a 3 tall plant and 1 is a dwarf plant. So for any of the 7 trait, you can write this way. Is this clear? This is law of segregation explained. Next one is the law of independent assortment. For law of independent assortment, you need to know about the dihybrid cross. Law of independent assortment. So here we need to know about the dihybrid cross. So what is a dihybrid cross? 2 characters are taken into consideration. Dihybrid cross. So what are the 2 characters that I will take? I will take the seed shape and seed color, seed shape and seed color. Okay. What is the seed shape? Round and wrinkled. And what is the seed color? Yellow and green. Is that clear? So now let us see. So we always have a homozygous parent, right? Homozygous parent. So this one is a round yellow seed and wrinkled green seed. So this is the parent. Gametes, if you see, it will form one kind of gamete and this will form another kind of gamete. No other possibilities. Okay. So now F1 formed in this case is capital R, small r, capital Y, small y. Remember the same alphabets, the capital letter written first, small letter written later, and the same rule has to be followed for the next. According to the law of independent assortment, the contrasting characters means what? The seed shape and the seed color. They will form and they will go into the gametes independent of each other. Am I clear? So the contrasting characters passes into the offspring without any influence of the other contrasting characters. That is a law of independent assortment. So this one is F1. So F1 is a heterozygous thing and it will always show the dominant trait. So it will be a round and yellow seed. Is that clear? Round and yellow seed. So this one is your F1. Okay, F1 there is no ratio as such because all the plants will be of same kind, displaying the law of dominance, that is the dominant trait will be expressed. Let's move to the F2 first. Now for F2 generation in the examination, the question is asked on the make a Punnett square for gametes and make the Punnett square for the offsprings, that is different. Now when I talk about the F2, capital R, capital Y, small I cross. So in this case, if you want to make a Punnett square for the gametes, so there are two things. Okay, so what will do the Punnett square for this, for the gametes? What are the different possible gametes formed? Since there are two traits that is round and wrinkled, have it round and wrinkled, yellow and green. So what are the gametes formed? RY. Follow the rule, right children? Write R first. YR is wrong. So R small y, small r, small y. You have to follow the same order, whichever order. You can write yellow first and follow it everywhere. So these are the gametes formed. So this gives rise to the gametes RY, R. Similar gametes are formed by this also. Okay, these gametes are formed by this, and the another one will also form the same gametes. Okay. Now for the F2, we have to draw a Punnett square. Okay, let me rub this quickly so that we can draw the Punnett square. Gametes you have understood? Let us move to the Punnett square. In that case, let us have the gametes RY, 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 RY. Similarly, do it with me everyone. Okay, I'll quickly fill in the box. The box is ready. Let us quickly find out. Now wherever you have got capital R and capital Y, that will give you a trait as round and yellow. Okay, round and yellow, that will be R and capital R and capital Y. Similarly, wherever you have small r's, that is small r, capital Y and anything, it is your wrinkled yellow. Okay, again you have got capital R, small I, that is your round and green, and the last one, that is your small r, y, that is your wrinkled and green. Okay, so in this case you will find out that capital R 1 2 3 4 5 6 7 8 9, there are 9 round yellow, 1, then you have got 2, and you have got 3 round and green, right, then 1 2 3 wrinkled and yellow, and 1 wrinkled and green. So this gives your phenotypic ratio of a dihybrid cross as 9:3:3:1. Please by heart it. 9:3:3:1. You know how you have got it, right? So this is very important for the exam, children. Is that clear? Next topic for this, which is important for the exam, is your sex linked inheritance. Sex linked inheritance is the inheritance of any disorder or character which is present exclusively on the X or Y chromosome. Okay, before talking about exclusively, this you have to write it. The allele should be exclusively on the X and Y chromosome, and if you just bifurcate it as X linked, then you have to write the allele of the inheritance, allele of the disease or the character must be present exclusively on X chromosome. Okay, so X linked inheritance, generally we study about hemophilia and color blindness. Color blindness, to differentiate between the colors, there is an issue we have already talked about in the sense organ chapter when we are talking about eyes, and hemophilia in the circulatory system when the blood fails to clot. Okay, these two are genetic disorders. In this case what happens, we don't write here capital letter and small letter the way we wrote for the pea plant. Okay, here we are talking about chromosome, we are not talking about alleles. So you have to write X for a healthy X chromosome, or you can write X dot for the chromosome which carries the alleles for the disorder. So you write X dot. If it is hemophilia, you can write XH. If it is color blindness, you can write XC. That is how we write. Okay. So now for understanding the pattern of inheritance, we have to first understand the sex determination in human beings. Sex determination in human beings generally depends upon the type of sperm that it fuses with the egg. So you know that sperm, that is the male gamete, has either 22 plus X chromosome or 22 plus Y chromosome. The ovum, that is the female gamete, has 22 plus X. So if the male and the female, that is 22 plus X, fuses with 22 plus X, gives rise to 44 plus XX, that is a female child. Yes, or in the other case, if 22 plus Y fuses with 22 plus X, it gives rise to 44 plus XY, that is the male child. Is this clear? So you can see that a son inherits Y chromosome from the father and X chromosome from the mother. The daughter has one father's X and one mother's X chromosome. Is that clear? So when you talk about the sex-linked inheritance, like you talk about a hemophilic father and a normal mother, right? This one is a hemophilic father. Hemophilic father. Father has no option beta in this case. Reason is the father will express hemophilia. It has got only one X chromosome, and if that one X chromosome contains a diseased allele, then the father will express, the son, the male will express. That is the reason why the males are more prone to X-linked diseases, because they have only one copy of X chromosome. In case of female, the chances of being carrier increases. Okay, you know that for female to be hemophilic, she has to have a hemophilic father and a carrier mother or a hemophilic mother. Chances are low, right? So this hemophilic father marries a normal female. I'll just draw a Punnett square, that will be easy to understand. X, H and X and Y, that is the father, and XX, right? So you can see here the father and the mother. So here the daughter becomes carrier. The daughter becomes carrier and the sons are healthy. Reason is the sons are healthy because the mother was healthy. So the X chromosome which the son has received, it has been received from the mother. So the son is healthy. But daughters are also carrier. They are also not displaying the disease. They are the carriers. Now next question is, this carrier daughter marries a normal man. So let me quickly rub this. Yes, so now the hemophilic, that is the carrier daughter, marries a normal man. Normal man means who does not have a disease. I am not talking about the mental abilities, children, I am talking about the disease. Kindly do not take me wrong. So here X and normal man, no? So XY and the mother X is a carrier. Okay, so the father is here and the mother. Now what happens? The first daughter is completely healthy. The second daughter is also healthy, but she is a carrier. The first son is normal, but the second child, the second possibility of a son is that the son will be a hemophilic son. Okay, so 50% of her son, there are 50% chance that the son born to this couple will be hemophilic, and there is 100% chance that the daughters will not be hemophilic, but the 50% of the daughters will be carriers, right? So in this case there was no one hemophilic. In this case the son is hemophilic. Now what happens? Let us have the carrier mother marries a hemophilic man. Okay, so what are the possibilities? Let us see. Children, Punnett square shows the possibilities. Pedigree shows the real offsprings, but Punnett square shows the possible combinations. Every time a couple will have 4 children, it's not possible. These are the possibilities. So in this case, you see, this is the father and the mother. So the first daughter, the first possibility is the carrier daughter. 50% of the daughters will be hemophilic. Son is one son is normal. 50% of the son is also hemophilic. So what is the possibility? This is hemophilic. This is hemophilic. So if you see overall, 50% of the children will be hemophilic. If you see only son, 50% of the son is hemophilic. 50% chance is there that the son will be hemophilic. 50% chance is there for the daughter to be hemophilic. Another one is a carrier daughter, but she is normal. She will not display hemophilia. And in this case, 50% the son is normal, 50% hemophilic. These type of questions can be asked in the examination. You have to guess the offspring and the possibility of them getting the disease. Is this clear? Any doubt? Now, as promised, as I have told you that we will discuss some important questions. The basic questions from this part of your textbook, that is Unit 1 Basic Biology, is around 10 to 12 marks. In that 10 to 12 marks, 5 marks of question on mitosis, the phases, identification of those phases is there. Then you have cross. Law of Mendel is going to be asked. The 3 laws are asked, it will be asked. So you have to be very thorough with the 3 laws of genetics. Is that clear? The Pisum sativum, the scientific name of the plant used by Mendel. The traits, sometimes in the question it's given what is dominant, what is recessive. Rarely it may not be given. You should know about the 7 traits that Mendel has studied. Is that clear? For any other doubts or query, put it in the comment section. I'll be happy to help. Happy learning, children. Have a great day.